if anyone has questions, please come use the microphone, and go ahead.
Yes, a couple of questions to Dr. Abernethy. Yes, Bernard Munoz from Faster Cures. There is over the last 3 decades, I've seen many attempts at trying to come up with a common template in order to accommodate databases that had been created and had never been meant to be interfaced. And the problem with that approach is that it is soon defeated by science. Science advances, and we discover new parameters that are worth measuring, so new biomarkers that weren't included in the original big template that was supposed to accommodate everything. Conversely, we find out that perhaps parameters that had been identified were confounded and are maybe the combination of various subparameters. And just following the common template approach leads to constantly restructuring the common template and is self-defeating. So that's my real-life observation. And conversely, we look at the scientists at Google, for example, or at the NSA. I mean, they constantly merge and extract knowledge out of unstructured data without the need for a common template. So are we perhaps making our lives more difficult than it needs to be by going after an elusive common template, or could we just emulate what's being done in outside the life sciences and basically find the answers that we need?
I love this question. So I'm going to divide it into 2 categories. So the first part of what you're asking is, how do we deal with the fact that essentially our understanding of clinical care and science is constantly evolving, and whatever common data model you generate is always going to be old news. And at least the approach that we've taken, and some of this was generated also from what I learned during my time at Duke when we were doing a number of Go grants and other things for the NCI, is to start with a parsimonious model, but hold on to the ability to go back in and add new data points as you need over time. And so at Flatiron, for example, we don't process or get everything right now, but instead do deep dives as necessary. And that falls into 2 categories. Sometimes it's to add in additional dimensions so that you can understand more about a particular cohort or story. So in the case of heart failure, we don't collect information about heart failure for every cancer patient, but instead zoom in when necessary. Or that now standard of care is updated and we need to start adding specific data sets into the large-scale data model. So for example, ROS1, we heard from Mace Rothenberg earlier this morning about ROS1, and so we've added ROS1 into our lung cancer data model because that's really much more a part of standard of care. And I think we've got the need to continue to have a flexibility of how we do that across time. And that's part 1. Part 2 of what you asked is, isn't this all silliness? And other industries have figured this out without this sort of expectation to get back to some kind of standardized data model that everybody can look to. And on one side, I think there is great excitement about artificial intelligence and machine learning that we all can get around. But I think we have a responsibility of being very careful about how we apply those approaches until we understand what they can work for and when. The challenge with, for example, artificial intelligence and machine learning is that it's hard to know when it starts to go off the rails in a direction that's not consistent with clinical decision making, and you don't have the ability to adjudicate and do so in a rational and hypothesis driven manner. And so one of the tasks here is to start to fold it in a careful way.
So that was great, and I'll give you a follow-up maybe for the whole panel to that. So how do you feel, what do you think about the intersection between the more conventional clinical trials level data gathering and what we're seeing from real-world evidence? I mean, what do you see about the interplay? And it's to anybody on the panel. Obviously, Maria, you spoke to this a lot. Where do we see the intersection coming in, and is this something we need to actively seek? Because we see the danger of, you go off and do your stuff there, the conventional clinical trials people go off and do their stuff here, and there's not the give and take that we're going to need to make both sides better. So I'll just throw it out there.
Let me take a stab at it. I think all sources of information are important, but they tell us different things. I think if we think we are getting the same answer from one source of data and the other, that's a problem. So the randomized clinical setting or any other clinical trial setting tells you if the drug works, first of all. So in a very clean environment, can you say actually this drug has some activity there or has an effect? Whereas in the real world, you're talking more about comparative effectiveness. You're looking at, okay, why is this physician making a choice of this particular product to this patient, but to some other patient, another set of treatment that's being chosen. So the two are answering very different things, but they complement each other. You do need both of them in the end. The real world data is more of the clinical practice, whereas the clinical trial data is more to see really if the drug works. Now, do we need that pristine environment at all for that? You can question, but you don't know much about the safety of the data when particularly you're looking at early clinical trials, and then to say, okay, this is already in the real world. So the real world gets it only after you have seen the data in the clinical trials. So they are certainly generalizing to the bigger public, and we need to know. And as Amy was pointing out, this is very useful in safety characterization. And also, we must remember that clinical trials have a shorter follow-up. So we don't have the long-term safety data at all. And that we are seeing even in immunotherapies, as you saw last week in the New York Times, what was being reported. So, we do have issues with that. So both of them are important. The other part is, it's not that we have not used historical control data or historical data. We have used them, and now we are sort of putting them as real world. They were coming from either big registries or real world data like this to look at the historical control. Where do we use them? Typically in very rare diseases, or when we see that this is an unmet medical need and we have to use that. And for the imatinib approval, some of the small subgroups, we did take case reports and use that for approval. So they all give different things, and they're all very useful, important, but they complement each other.
So how I look at it, and I agree with all this, but how I look at it is that the real world data are more about the precision of the data that you can get from real world data, which means they are imprecise compared to the clinical trial data for several reasons. One, because the endpoint is unclear, because we are so used to the endpoint of measuring PFS every 2 months and comparing this between the arms, which eventually, if a hazard ratio is 0.5 in some sort of comparison, it really doesn't matter in the big picture, I think, whether the hazard ratio is 0.56 or 0.42 in real world. What matters is that there is a humongous difference. And I think that doing a real world study comparing gemcitabine with carboplatin is probably not the right thing to do, because you cannot really show anything. However, comparing, agreeing to a real-world pragmatic study, if you want to have additional comparator for a drug that has hazard ratio of 0.5 is probably fine. So I truly believe that it is about the endpoint that you can show, just like Amy said, that in the records there is several date of death, and which one, so you don't have the precision when you collect it in a clinical trial.
Can I?
Yeah.
Oh, sorry.
I was just gonna comment that I think that that as you nicely described, Amy, that we can use real-world data to evaluate, say, for example, eligibility criteria that we're using on clinical trials and potentially modify clinical trial designs in the future such that we can expand our eligibility criteria. And so I think it's sort of an iterative approach, perhaps, that we can use to complement each other.
I really just echo that. Most of the questions I've been getting now when I'm wearing my NIH hat or my PCORI hat are about just that. It's trying to plan trials, plan enrollment criteria, try different enrollment criteria for trials. That's really what people have been trying to use the data for. Other than, I mean, there's the other piece of comparative safety, comparative effectiveness, but trial design has been one of the kind of the top recently, kind of the requests we're getting.
Hi, Craig Tendler from Janssen. So a question about the example you gave on the Kadcyla project. You mentioned the importance of outlining study objectives, and I just wonder if there was a pre-specification of a certain threshold for cardiotoxicity, and if that was designed in the project prospectively, do you think that could have at all had any impact on the black box warning? I'm just wondering, how did the 66 patients, was it just search the database and see what you get, or was it part of a prospective, analysis plan to say, we want to see if we can rule out in the patients with low ejection fraction, less than 10% incidence of cardiotoxicity?
So we were working through potential case examples that met the kind of criteria that we were trying to get to around what are important use cases for real-world evidence. So first of all, that's kind of where we started. In terms of pre-specifying the eligibility criteria, we started off with saying these need to be metastatic breast cancer patients who'd received Kadcyla within the time frame and who had EF of an EF of less than or equal to 50%. So systematically screened the entire data set for every patient that met those pre-specified criteria.
Okay, but there was no pre-specified statistical objective that we'd like to see if we can rule out a certain lower boundary for cardiac.
In this particular case, we did not have a power calculation, no.
Okay, and maybe a question for Dr. Sridhara. If, for example, that was the case, and the analysis plan included, some of this type of robust pre-specified analyses, do you envision the future that could potentially have an effect on labeling or black box warnings?
So for safety issues, yeah, I mean it can. See, what you're asking is the pragmatic clinical trials. We are still talking about using the sources that are there, the community as a whole, rather than specialized centers. But it is still a prospective study. We do prospective observational studies, and similarly, this is essentially a prospective observational study. In this case, what you're doing is you're randomizing the physicians rather than the patients. And depending on whatever is that physician's liking or treatment practice, you do that. And that was what was done in the GSK study. So you can do it and you will get some of the answers. But in the case of oncology, for example, what is the endpoint can we look for is going to be very different from what we can get in randomized clinical trials. We cannot really go for progression-free survival or tumor response rate because RECIST is a criteria that's used by specialists. It's not going to be there in every community hospital. And neither, you may not get even the overall survival for that matter. Yes, with a lot of probing, you may get the death date from the death registry, but you may not even have that. The follow-up is an issue. You may have, the patient may be going to a particular doctor, but then may shift to somebody else, and the record may just go off the record from that. So the formal follow-up that you see in clinical trials aren't going to be there to get some of these endpoints. So it may be just time to a treatment change. A change of treatment could be the thing that you may want to see in those. So that's why they are answering different things, but valid things.
Mark Retain, Chicago. I think there's one example that I'm familiar with that's proof of concept for real-world evidence, and that's the Vanderbilt experience with BioView. So BioView is a database at Vanderbilt where they basically took a large cohort of patients measured in the tens of thousands, did genome-wide genotyping, and took the EMR and digitalized it with all the ICD-9 codes, I believe. And so you basically have this large database that has diagnosis codes and has genome-wide genotyping. And they've used it both for discovery in an effort that they refer to as PheWAS, where you have your favorite polymorphism. You can see what phenotypes associated with it. And they've also used it for replication, where you do a more conventional genome-wide association study and then go into their database and you're able to replicate. It's really fascinating, and it's probably worth studying, but it brings to the point that the area where this may be most useful is around biomarkers, where patients who are getting the same therapies, but for whom we have good biomarker data, can be analyzed and compared because there's proof of concept that we can both discover and validate biomarkers using real-world evidence.
So one of the data sets that we maintain right now is 20,000 plus patients. That's a combination of the complete EMR data coupled with their Foundation Medicine NGS reports, including all the VCF files. We're also finding that many academic institutions want to use their processed EHR data to act as clinical annotation, to go along with their biorepository, et cetera. And you know, this helps both on the biologic discovery side. It helps in understanding and identifying biomarkers. And I also think that the other place that we're seeing a substantial number of use cases is along the lines of what Jeff just described, which is on the study design side. Because now you've got a lot of the key features that help you pressure test, for example, eligibility criteria and how to better design your studies.
So what you said is true, you know, in terms of learning about the history of the disease or knowing more about the disease itself. You definitely can use all of this data to tell us more about it, and it will help us in future clinical trials what we should be targeting or how we can run the studies, what should be the control. Yeah.
But you can also pick up adverse drug reactions that are associated.
Yeah,
you know, I mean, there's all kinds of things one
Safety
can pick.
for certain.
And, you know, they did it primarily with a drug-based, you know, context. That was the primary goal originally.
Well, I'd like to thank the panel for a very exciting session, and Dr. Shilsky.
So I'm not going to prolong what's already been a long day with any lengthy remarks tonight. I think we've had a terrific day, very interesting, very productive. Hopefully lots of good thoughts are flowing. We will reconvene tomorrow morning for breakfast at 7:30, and then our session will start promptly at 8 tomorrow. We will try to have some kind of a synthesis to present to you as part of the wrap-up before we conclude tomorrow afternoon. So have a good evening, and we'll see you tomorrow.